or like AMP, for example, is another association meeting. They have websites and stuff like that.
Mm-hmm.
So, yeah, people go to websites. But people who are in the molecular diagnostics space also read a lot of papers. A lot of that is very, research focused. A lot of, assays that are being developed in a research setting. That's true in the immuno-oncology space right now. Everybody and their grandmother is trying to figure out what are better immunomonitoring assays, given the PD-L1 diagnostic sort of disaster, quote unquote. I'm not saying it's bad. It's just, it's not as predictive as one would like. And so the race is on, and a lot of people do this in their labs, and of course a lot of that is ultimately translated then into actual diagnostic assays. So people who are in that space are very closely tied to the academic communities in which the research is happening.
You just sparked something once again when you talked about the PD-L1 experience. Can you just, you don't have to name any pharmaceutical names or companies or anything, but if there's why that was not a good experience for the pathology community, could you talk a little bit about that experience of why that wouldn't be a good, yeah.
Well, I think when you look at how it was developed, it was not so much engagement of the academic pathology community and whether they thought this was a good assay or whether, the cutoffs and all of that, the ridiculous. Like a 1% positivity rate with any of these, crappy IHC stains, you can get that positivity rate. But for whatever reason in the original clinical trial and then later in the FDA approval for some of the drugs, these cutoffs were put in. And I assume that, these assays were developed jointly with a CRO, and so they just provided whatever, the pharma told CRO they wanted to have. So it was a bad assay to start with. The biology is very weak too, and there's certainly better ways to measure this. But again, pathologists were not involved, and then the whole wave came, and then people said, well, you need to implement the assay, especially if you go to a private lab or a smaller, even academic laboratory. You have to develop an assay that people know doesn't really work well. Each drug was approved with its own antibody, sometimes its own platform, its own hardware platform, which was often older, outdated ones. So to officially, for example, be FDA compliant with the assay, you would have to not just buy the antibody, but you have to buy the instrument. So who would buy an instrument that they otherwise would never use? And then you have to understand so that the financials also in many hospitals where pathology then basically would have to come up with the finances to buy the instrument, et cetera. Not a clear way to get reimbursed for the assay either, just to make sure a few patients get on this drug at this medical center. So people in big centers got around it and said, look, we don't give a damn, basically. We just evaluate and validate one of those antibodies, the one we think works the best. We're going to use that for all of those IO class drugs. We basically don't really follow the FDA approval. We just use a lab-developed test. We're not going to develop one antibody assay for each drug. We're going to run it on our own platform. But that's sometimes prohibitive and difficult to do for smaller labs. So this entire scenario where, an assay was approved as a companion diagnostic that is just tied to one drug, but it's so different from the competitor's drug, the assay itself, all of that creates an environment that's not very attractive for pathologists to be excited about it. And then given also that the science behind the assays is kind of a little shaky and the cutoffs and all of that is very qualitative and not very reproducible and so forth. So overall, basically it's a poster child of, I guess, how you don't want to do it. And I'm not blaming one single company. I think it's overall, it's the field that hasn't done well. And what it means, the drugs work, don't get me wrong. It's just we don't know who will really respond well. So there are obviously better assays, and so the lesson is that the earlier you engage, especially key opinion leaders and pathologists who are in very active research environments and really at the forefront of their discipline, I think they should be just as much engaged as medical oncologists or others who ultimately help drive the phase 1 trial forward and identify the right cohort of patients where the drug works. And I think that's going to be, a lesson for the near future is how do we do a much better job of, developing companion diagnostics that have wide acceptance, that people both